Hey everyone, and welcome to this explainer. Okay, imagine a world where the physical limitations of scientific training, the thousands of dollars wasted on single-use materials, the hazardous chemical spills that literally clear out entire buildings, the constant scheduling conflicts, what if all of that was just completely a thing of the past? Well, today, we're stepping into an era where cutting-edge tech meets critical medical science, and it is absolutely reshaping how the next generation of medical professionals learns. For the longest time, mastering complex pharmacology meant you needed massive physical resources. We're talking immense safety protocols to protect students from hazardous materials and that constant, strict over-the-shoulder supervision. But today, the modern digital gateway is completely bypassing that old bottleneck. We're moving away from those expensive, high-risk physical labs and diving straight into incredibly realistic, entirely risk-free digital environments. It is a real game changer. So, let's make sure we're on the same page here and define exactly what this tech actually is. A virtual pharmacology lab is essentially a computer-based environment that perfectly replicates a real laboratory. It lets students conduct drug experiments, test tissue samples, and analyze complex data entirely in a digital space. But here's the absolute biggest takeaway. It completely eliminates the need for physical equipment, and, crucially, it removes the need for animal testing. It is a 100% safe, completely risk-free environment. Here is our roadmap for today. We'll cover the virtual lab revolution, why virtual simulations work, mastering core pharmacology concepts, real experiments in a virtual world, and finally, the future of training. All right, Section 1. The virtual lab revolution, breaking the physical boundaries. The contrast between these two approaches is just staggering. On one side, in a physical lab, you're inherently dealing with risks to patient safety, hazardous materials, and heavily limited access. Because, let's face it, universities only have so much physical space and funding, right? Virtual labs flip this whole dynamic on its head. They are 100% safe, they're incredibly cost-effective since you aren't constantly repurchasing single-use materials or mopping up accidental spills, and honestly, the best part? They are accessible anytime, anywhere. Students can repeat these simulations as many times as they need to, getting that true self-paced learning experience without burning through a single drop of valuable lab resources. But, it's easy to just call this cool tech. Does it actually improve education? Let's look at Section 2, why virtual simulations work. The proof is in the data. So we aren't just guessing here. When surveying actual medical students across 10 key areas of virtual pharmacology experiments, the data was incredibly clear. Looking at pre-test and post-test evaluations, there wasn't just a slight bump. There was a marked, significant improvement in their understanding of complex drug mechanisms. Because they could repeat experiments without that awful, dreaded fear of breaking expensive glassware, students strongly agreed that the extra practice was immensely beneficial. Plus, their anxiety about equipment challenges, that dropped substantially. Ultimately, these virtual labs weren't just seen as some neat digital trick. They were recognized as a highly powerful aid in building actual, real-world clinical skills. Moving right along to Section 3, Mastering Core Pharmacology Concepts, Visualizing the Invisible. If you know pharmacology, you know ADME. It stands for absorption, distribution, metabolism, and excretion. It's the fundamental study of exactly how drugs move through and ultimately affect the human body. Now, if you're just reading a textbook, this is incredibly dense, tough material to grasp. But in a virtual lab, by using interactive 3D simulations, students can actually visualize these invisible biological mechanisms. It's like turning on the lights in a dark room. They can see the physiological responses happen right before their eyes in a dynamic, perfectly controlled setting. And it's not just ADME either. These platforms let students dive deep into pharmacokinetics and pharmacodynamics, which is basically studying how drug concentration influences therapeutic and toxic effects. They can visualize the actual mechanisms of drug action, like seeing how a medication interacts with specific cellular receptors and enzymes. They can safely simulate toxicology and adverse drug reactions without any risk to a living subject. They can even tackle antimicrobial therapy, running rigorous virtual antibiotic sensitivity tests to see exactly how susceptible a highly resistant microbe is to different treatments. Okay, Section 4, Real Experiments in a Virtual World. Let's look at a case study of PraxiLabs. PraxiLabs is a fantastic platform that takes all this theoretical curriculum and brings it to life. Because it's cloud-based and highly interactive, students get instant guidance, real-time performance tracking, and immediate evaluation the second they work through a procedure. Let's actually walk through a few of the specific scientific quests they can take on. Picture this. You're a student tasked with an in vitro viability assay using XTT. This isn't a reading assignment. It is a full-on mission. You're testing how well-cultured cells survive after being exposed to varying concentrations of nanoparticles. Step 1, you virtually dilute the cells to the right count for seeding in a 96-well plate. Next up, you calculate and prep the exact geometric concentrations of the nanoparticle doses in the culture media. Then, you add the tetrazolium salt XTT solution to the cells. You actually operate the virtual microplate reader yourself to scan the assay, and finally, you calculate the exact viability percentage of those exposed cells. You are doing the math, you're handling the virtual instruments, and you are uncovering the results firsthand. Or, let's look at the Annex NV assay. To cut through the heavy medical jargon for a second, this is basically a visual quest to identify necrotic and apoptotic or dying cells. First, you treat your virtual cells with cytotoxic agents. Then, you harvest them using a very specific binding buffer containing Annex NV and propydium iodide. From there, you basically step into the role of microscopic detective, analyzing the cells thoroughly using a virtual fluorescent microscope. And you don't just look at them, you graphically represent and interpret your findings using dot plots. It is the rigorous, step-by-step scientific method executed flawlessly on a computer. But hey, it's not all just flashy tests, right? It's also about learning fundamental laboratory discipline. In the adherent cell culturing module for mammalian cell lines, students have to apply strict aseptic techniques following good laboratory practice guidelines to the letter. They learn how to meticulously handle the instruments needed to propagate and subculture cells, getting them ready for future assays. They even learn the exact protocol for freezing cells in virtual liquid nitrogen for long-term storage and how to properly thaw them for seeding. They are internalizing these crucial physical safety standards without ever actually being in physical danger. Finally, Section 5, the future of training, shaping tomorrow's healthcare. So I want to leave you with this thought. What happens to global healthcare when every single medical professional logs thousands of hours of completely risk-free practice before they ever even touch a human patient? Think about that. When trial and error doesn't cost thousands of dollars in materials or worse, risk human safety. That is the ultimate promise of the Virtual Pharmacology Lab. It is making high-quality, rigorous medical education accessible, repeatable, and scalable in a way we have literally never seen before. It's a profound shift, and it's going to be absolutely fascinating to watch how this elevates the standard of care worldwide. Thank you so much for joining me on this explainer. I hope this sparked your curiosity, and as always, keep learning.